welcome to the March edition of the Liver Seminars Young Investigators Edition. So on behalf of Malu Martinez Chantar, the PI of our laboratory, and myself, Teresa Cardozo, I would like to acknowledge, first of all, the organizers of Liver Seminars, Jordi Gracia and Rafael Bañares, for this opportunity. We are very excited to present our speaker today. We have we have Sergio Rodriguez Cuenca. Is a senior research assistant at the Wellcome Trust MRC Institute of Metabolic Science from the University of Cambridge. Sergio obtained his bachelor degree in biology and biochemistry at the University of Balearic Islands in Spain. Following its postgraduate program, which focused on the role of sexual dimorphism in the energy metabolism of white and brown adipose tissue, he defended his PhD thesis in 2005. In 2006, Sergio was awarded a Marie Curie Fellowship and joined Professor Vidal Puig's lab at the Department of Clinical Biochemistry in Cambridge, UK, where from 2008 to 2013, he has worked as a research associate. From 2020, Sergio became affiliated to the Cambridge Nanjing Center of Technology and Innovation in China. So Sergio's current areas of interest include the qualitative aspects of glycerophospholipids and sphingolipids remodeling and their relevance in the context of obesity, diabetes, and NAFLD. Also, the role of mitochondrial dysfunction in the development of insulin resistance or other metabolic disturbances that are associated with obesity. And also is very interested in peroxisome biology in the context of obesity. Today, Sergio will tell us about the role of phosphatidyl inositol remodeling to mediate dietary effects on fatty liver. At the end of this talk, Sergio will be glad to answer your questions that you can either write on the chat or raise your hand if you want to ask yourself. And thank you very much, Sergio, for accepting our invitation. And whenever you are ready, you can start. Thank you.
Teresa, thank you very much for this kind introduction. I would like to thank the organizers of this event, give the opportunity to present. For the ones of you that they don't know us, just to summarize briefly the 5 different areas of research that we are currently undertaking in Cambridge. The first theme relates to the adipose tissue biology, especially in the context of obesity and type 2 diabetes. The second area of research relates to the activation of thermogenic programs, both in brown and muscle, in the context of energy expenditure. We are also developing a theme number 3 related to lipotoxicity and how the qualitative changes in the lipid composition affects the comorbidities associated to obesity, with special attention to fatty liver and cardiovascular diseases. We also have 2 additional programs related to macrophage biology and inflammation, once again, in the context of obesity, diabetes, and associated comorbidities. And the last thing relates to an stem cell program when we are aiming to obtain functional brown and white human cells. Our motto at TBP Lab is based in an adipocentric point of view, where basically we believe that the failure of the adipose tissue to expand in the context of obesity, so in the response of a positive energy balance, or in cases of a lipodystrophy, will cause the ectopic accumulation of lipids in peripheral organs, including the heart, pancreas, skeletal muscle, as well as liver. We believe that this accumulation of ectopic lipids will contribute to both the onset and the worsening of the comorbidities associated to obesity, including NAFLD. It is well known that the accumulation of triglycerides is a feature of steatosis. Nevertheless, we believe that this might not be the main pathogenic factor in the evolution to NASH. We believe that the pathogenic potential of lipids due to lipids-induced toxicity is determined mainly by the biochemical properties of those lipids, as well as all the allostatic responses that are derived from changes in the qualitative and both quantitative composition. To answer the question of how changes in the qualitative composition of lipids might contribute to these comorbidities associated to obesity, including NAFLD, we have developed an extensive program of research focused in altering metabolic processes related to the biosynthesis of fatty acids, like unsaturation or elongation, as well as targeting specific genes involved in the biosynthesis or remodeling of particular group of lipids, including ceramides, phosphatidylcholine, plasmalogens, cholesterol, and particularly the remodeling of phosphatidylcholine, ethanolamines, and phosphatidylinositols. And today I'm going to be focusing part of my talk to the project related to ENVO7. What is ENVO7? ENVO7 is one of the enzymes that participates in the LAN cycle. The main function is the remodeling of phosphatidylinositol lipids. And this is happening through the concerted action of two different enzymes. First of all, phospholipase A2 will remove the fatty acid moiety located in the fatty acid, and this will be substituted by ENVO7 with arachidonic acid that will be previously activated with ACSL enzyme. We believe that perturbations in both the activity of the expression of ENVO7 as well as the other members of the LAN cycle will contribute to the metabolic syndrome by altering the metabolism of both adipocytes, hepatocytes, as well as the inflammatory response of the immune cells through very basic cellular processes. A, altering the biophysical properties of membranes, B, altering the recycling of polyunsaturated fatty acids, or B, altering signaling events. In particular, ENVO7 will be driving alterations in the phosphatidylinositol phosphate signal. The clinical relevance of ENVO7 came from these two papers. The one on the left side provides the first evidence showing that a particular mutation in ENVO7 was associated to an increased risk for alcoholic-related cirrhosis. Soon after, the laboratory of Stefano Romeo in Göteborg provide consistent evidence that the same mutation was associated to a high prevalence of steatosis, necroinflammation, as well as fibrosis. And this was happening around 2016. But soon after, there was a plethora of different groups of research that provide similar evidence, where this particular mutation associated with increased risk of liver inflammation, fibrosis, liver damage, and so on. Strengthened the concept that the decreased expression of ENVO7 caused by this particular mutation was somehow an increased pathogenic factor in the evolution of NAFLD. Nevertheless, in the same number of years, there was a number of different papers that they couldn't provide this kind of association. Despite this could be easily explained by power calculation difference or sample size, there is a third unexplored possibility that consists of in the potential interaction of the genotype and the diet. In order to answer this question, in the case of the ENVO7, we develop as a part of the EPUS consortium an extensive nutritional challenge where basically we have been using different nutritional interventions to force the NAFLD event. We have been using both the classical obesogenic diet, 45% coming from the kilocalories, as well as the American lifestyle obese-induced diet, the Alios diet, both in absence and in presence of fructose and glucose, as well as the classical Western diet plus fructose and glucose, and then two additional models for validation purposes. We use, for this purpose, we have used the heterozygote, the global heterozygote for ENVO7, as we consider this to better recapitulate the decreased expression of this gene described for the carriers of this mutation, but most importantly, to mimic any potential allostatic responses triggered to maintain metabolic stability. This is a concept that we are particularly interested in. Before embarking on the phenotypical analysis of our animal model, something that we did is that explore how ENVO7 was altered in different data sets that we have available in the lab. Just to cut the story short, basically, we didn't see any substantial effect in the expression of ENVO7 in liver, gonadal adipose tissue, or inguinal when we were using classical obesogenic diets like 45%. Nevertheless, we start seeing a dramatic reduction in the expression of ENVO7 only in conditions of severe obesogenic diets, either half and 60%, or in genetically models of obesity, like the OBOB mice. Also of interest was the fact that in a transient episode between fasting and refed, we could also see a reduction in the expression levels of ENVO7. This was perfectly in line with recent publication from Luca Valenti lab last year, where basically they show that in models of hyperinsulinemia, the expression of ENVO7 was dramatically reduced. They also provide very elegant experiments in vivo where they show basically that the treatment with insulin reduced the expression of both the RNA and the protein levels of ENVO7 through a mechanism that was dependent of PI3 kinase, as you can see here. Early last year, just before the lockdown, Helsley and colleagues reported the first preclinical evidence showing that the ablation of ENVO7 using an antisense oligomer was directly associated with the promotion of NAFLD. Also, these animals they were showing a substantial increase in inflammatory and fibrosis signature, impaired glucose tolerant tests, and show dramatic change in hyperinsulinemia and severe impairment in the insulin action in the liver. They also provide some evidence supporting the notion that lysophosphatidylate inositols that will be substantially increased when the MBO7 is absent are sufficient to promote fibroinflammation in the liver of these animals. Nevertheless, despite they use an antisense oligomer and the reduction of MBO7 was reduced in the adipose tissue of these animals, they couldn't see any changes in the transcriptome of these animals. Soon after, 4 different labs in Europe, States, and Japan provide also evidence of how the ablation of MBO7 were contributing to an increase in the steatosis of these animals, increased liver damage, as well as increased fibrosis. Nevertheless, there are some inconsistencies regarding the association of increased inflammation and fibrosis in these animal models, but this is probably substantially dependent on the animal models used. In particular, most of these groups has been using hepatocyte-specific knockout models. At this stage, 2 of these labs provide some potential mechanisms that can explain how the ablation of MBO7 could increase the accumulation of triglycerides in the liver. The first one, that was provided by Xia in 2020, just should provide strong evidence where in the MBO7 knockout animals, there was a dramatic activation and cleavage of a SREP1 protein, promoting de novo lipogenesis. And this was totally planned when they were combining the MBO7 animal with the SCAP protein. That is one of the first activators of SREP. The second mechanism that is more intriguing is the one provided by Tanaka and Lucas Valenti's lab, where basically they provide the strong evidence of the existence of a non-canonical biosynthesis a non-canonical biosynthesis of triglycerides by the increase in the phosphatidylate inositol turnover. Basically, they suggest that the reduction in the levels of phosphatidylate inositol containing arachidonic acid will promote the biosynthesis of phosphatidylate inositol by activating the CDP synthase enzyme, and at the same time that they are activating a phospholipase C enzyme, although they haven't detected which one it is. This will render a substantial increase in the levels of the acylglycerol that will fuel the biosynthesis of triglycerides. Interestingly, any of those mechanisms are mutually exclusive, so probably both systems are really happening. But what happened in our animal models? When we were exploring the effect of our different diets, the first thing that we observed is that only in the case of the animals fed allulose, fructose, and glucose, we saw a significant increase in the size of the liver of these animals. This is something that we didn't observe when we were providing the animals with a classical allulose or a classical obesogenic diet. This increase in the size of the liver was accompanied by an increase in markers of liver damage, ALT and AST, as well as an increase in the steatotic area of the liver of these animals. Surprisingly to us, something that we consider paradoxically is despite the animals fed half-fat diet 45% didn't show any evidence in altering the size of the liver or difference in the steatotic area, the levels of the liver enzymes were significantly reduced, providing the first evidence of a dietary specific effect. It won't be a surprise to any of you that basically any of the diets that we have used, with the exception of the Western diet fructose glucose, provide histological detectable fibrosis. But even in this case, we didn't observe any sign, any statistical difference between the genotypes. But what happened to the fat percentage and the body weight of these animals? So we didn't observe any dramatical difference in the fat percentage of the animals, independently of the nutritional intervention used, with the exception of the animals fed Western diet, where basically, despite the total fat percentage of the animals were no different, the size of the inguinal depot and the retroperitoneal depot was substantially decreased, with no difference in the levels of gonadal fat. Our current interpretation at the moment is that this is reminiscence of a lipodystrophic-like phenotype, and we are currently investigating the causes of this. when we were exploring the serum biochemistry of these animals, we couldn't find any dramatic changes in the levels of triglycerides, of cholesterol, with just a marginal increase in the level of cholesterol when the animals were fed the allulose, fructose, and glucose diet. We also observed a statistical significant decrease in the level of cholesterol when the animals were fed the Western diet fructose glucose. Despite this marginal phenotype that we have observed, we decided to go ahead and go for a transcriptomic analysis of the liver of these animals. And basically, we were choosing the models where we have some evidence of liver interaction. As you can see here, we have been dividing the pathway analysis in different sections: damage, inflammation, and fibrosis, and upstream regulators. As you can see here, the MBO7 animals that will fare exclusively with the HFAT diet with the allulose, fructose, and glucose, they have a significant increase in pathways suggesting liver damage. Opposite, when the MBO7 animal were fed the classical obesogenic diet, the HFAT diet 45%, we have a reduction in the expression of the genes and pathways, evidencing probably some kind of protection effect, or at least a totally opposed phenotype. This was also observed in pathways related to both inflammation and fibrosis, where basically, once again, we have a significant increase in the MBO7 animals fed allulose, fructose, and glucose, and the opposite fingerprint when the animals were fed HFAT diet, where the animals fed the allulose without the fructose and glucose were staying somewhere in the middle. Interestingly, when we were exploring the upstream regulators, something that we observed is that in the animals, once again fed the allulose, fructose, and glucose, we have a very dramatic activation of SREP1, SREP2, as well as PPAR gamma, somehow suggesting an activation of de novo lipogenesis and cholesterol. This is going in line with the publications of Shia, showing the activation of SREP as one of the potential pathways explaining the increase in the de novo lipogenesis. Further analysis of the transcriptome of these animals reveal unique and sometimes opposed fingerprint in the MBO7 animals, depending on the nutritional intervention used. Here on the left side of the slide, we have just a summary of some of the pathways that were mostly dysregulated. Some of them were related to cellular proliferation, extracellular matrix and cytoskeleton, fatty acid metabolism, inflammation, as well as peroxisomal biology and OXPHOS. Given our interest in mitochondrial dysfunction, we decided to isolate the mitochondria of these animal models and perform a very basic analysis to analyze how well these mitochondria were performing in the different nutritional interventions. Just to cut the story short, basically we were using succinate, the substrate succinate in the presence of rotenone to establish the respiration driven mostly by the complex. And something that we observed, once again, it was some kind of dietary effect, where basically MBO7 animals fed HFAT diet 45% were showing a reduction in the respiratory control rate, somehow suggested an impairment in the mitochondrial performance in the liver of these animals. The opposite fingerprint was observed when we were feeding the animals with the allulose, fructose, and glucose. Somehow, this increase in the respiratory control rate is paradoxically suggesting an increase in the respiratory capacity and coupling. Western diet-fed animals were showing, similarly to the HFAT diet 45%, a decrease in the mitochondrial performance. One of our current hypotheses is that qualitative changes in the lipid composition due to the different nutritional challenges somehow could affect the action of MBO7 impairment as a pathogenic risk in the onset and development of NAFLD. So basically, one of the first things that we did is that using discriminant analysis is to prove that the lipid composition of the livers of these animals were able to discriminate the different groups. Here you can see that we were using both the global lipidome as well as other lipid fractions, and I'm going to talk about this. The different groups were clustering differently, especially the controlled diets versus the allulose-fed animals, and these were differing significantly between the chow and as well as the animals fed HFAT diet 45%. So basically, the next question is that how different is the lipidomics of these animals? And basically, we obtained a huge number of significant individual entities that were dysregulated across the different diets. But just to summarize briefly some of the findings, we were trying to find common patterns of dysregulation or changes that will somehow suggest a genotype per diet interaction. Basically, we didn't see, in the case of the triglyceride fraction, any major shift in the composition of the triglycerides. But basically, when we were establishing a correlation matrix using the fold change between the heterozygotes and the wild types, and the content of carbons in the triglycerides or the number of insaturations, something what we observe is a decrease in the number of triglycerides containing both unsaturated fatty acids or long fatty acids. And this was almost independently of any nutritional intervention use. So we also perform a comparison analysis of the different lipid species. And once again, we observe many changes in individual entities. But once again, we were most interested in the global picture. Something that we observed when we were analyzing both the phosphatidylethanolamin and phosphatidylcholine is that there was a differential effect when the animals were given both chow and half-fat diet 45% from where the animals were given allulose, fructose, and glucose, somehow suggesting that these diets were behaving differently and affecting the lipidome of the EMBO7 animals in a different manner. This is something that we also observed when we were exploring the phosphatidylcholine pool. So what happened with the lysophosphatidyl-cholines and lysophosphatidylethanolamin species? So basically, we also observed a totally opposed fingerprint regarding the signature of lysophosphatidyl-cholines. Here we can see in the heat map that we have an increase when the animals were fed chow and half-fat diet 5%, or a reduction when the animals were fed allulose, fructose, and glucose. Interestingly, something that we observe as a commonality between all these diets in relation to EMBO7 ablation, it was an increase in the levels of both ceramides and sphingomyelins, somehow suggesting that this is a common feature of the EMBO7 mice. Something that particularly interesting, because we have other projects active in the lab, it was the ablation of EMBO7 was only producing a decrease in the signature related to ether lipids of plasmalogens only exclusively when the animals were fed allulose, fructose, and glucose. Our next question was to try to identify whether we could discriminate both the EMBO7 heterozygote mice from their wild-type counterparts by a unique and particular lipid fingerprint. So basically, we were running some discriminant analysis to try to identify this lipid signature that could identify EMBO7 animals. Here you can see a summary of the different lipid species that are contributed mostly to differentiate EMBO7 animal from the wild-type counterpart in the different nutritional interventions. As you can see, the repertoire of lipids differs substantially between the different nutritional challenges. So basically, this is telling us, or we are interpreting this finding as diet being an important modifier when assessing how the impairment of the EMBO7 affects the hepatic lipidome. So going further with the metabolic profiling of our animals, and in line with what has been already published, we try to establish what happened to our animals. So basically, when we were conducting some insulin-tolerant tests, we couldn't notice any particular difference. So the animals were not showing any sign of insulin resistance in any of the nutritional interventions that we were testing. Nevertheless, when we were running a glucose-tolerant test, something that we observed is that EMBO7 animals, independently of if they were fed chow diet, half-fat diet 45%, or the allulose, fructose, and glucose, they were showing evident signs of a hyperglycemia. And this is something that we couldn't recapitulate in the context of the Western diet. When we were checking whether this was as a consequence of changes in the insulin levels, so we were analyzing the levels of fasting insulin. The only thing that we observed is that for the group that were fed allulose, fructose, and glucose, it was a significant increase in the levels of fasting insulin, something that we couldn't recapitulate for any of the other interventions. Intrigued by these findings, some of the findings that were provided in the previous papers regarding hyperinsulinemia, we were wondering whether pancreas was also somehow contributing to the overall phenotype of the animal. And basically what we did here, it was to run glucose stimulant insulin secretion experiment using the isolated pancreatic islets from these animals. And despite we couldn't see any genotype difference when we were stimulating these islets with a very high dose of glucose that we were equivalent to hyperglycemic status, we observed a very significant difference at the low concentrations of glucose. Okay? Basically, in the way that we are interpreting this result, comes from the fact that phosphatidylinositol phosphates are known to stimulate the KATP channel, repressing the secretion of insulin. Has been already established by some of our collaborators working in Braverham Institute that the ablation of MBO7 is causing a reduction in the global levels of phosphatidylinositol phosphates. So basically, is a justification that we are still trying to confirm further is that low levels of phosphatidylinositol phosphates could favor the closure of the KATP channels and increase an insulin leakage. Somehow this is suggesting that we should consider, when analyzing the global impact of the MBO7 ablation, more tissues are part of the liver. Okay. Given the results that were previously provided by other labs suggesting that the ablation of MBO7 in the hepatocytes were not associated with alterations in the secretions of VLDLs, we wanted to assess the whole body lipid homeostasis. So basically, we were running a lipid tolerant test. Okay? By oral gavage, we were giving the animals a bolus of olive oil. And basically, something that we observed is that in the case of the MBO7 animals in red, the animals were less deficient in clearing the lipid bolus. So you can see here an increase in the level of triglycerides over time, and as well as an increase in the level of free fatty acids. Our current interpretation, given that previous papers are not seeing any different in the secretion of VLDLs, is that we are interpreting this as a potential failure in the adipose tissue, although we cannot discard muscle either, regarding the capacity of uptaking and restrifying free fatty acids. If this is true, and this is something that we are working on, this could contribute to increase the flux of fatty acids toward liver of these animals. So one of our questions, because we have a particular interest in the adipose tissue, was basically to see what happened in the adipose tissue of these animals, okay? Given that our animal model is a global heterozygote. So interestingly, something that we observed is that in the MBO7 animals, when they were fed half-fat diet 45%, we have a significant increase in pathways related to TCA cycle and beta oxidation. And this is something that was not recapitulated when the animals were fed allele fructose and glucose. Moreover, we have also observed different particular and unique signatures depending on the nutritional intervention used. Interestingly, when the animals were fed only the chow and the classical chow diet, we observed a reduction in genes related to OXPHOS, somehow suggesting an impact of MBO7 ablation on mitochondrial dynamics, also in the adipose tissue of these animals. This is something that, once again, we are trying to elucidate further. Also of interest, when the MBO7 animals were fed the classical half-fat diet 45%, we also see an increase in genes related to fatty acid metabolism and elongation, as well as the biosynthesis of polyunsaturated fatty acids. Moreover, we also observe, in the case of the animals fed, once again, half-fat diet 45%, an increase in the signals related to pathways related to proliferation. Given these results, we were thinking that maybe we should pay a little bit of attention to what happened to the adipose tissue of our animals. And basically, we came back to this histological analysis. And despite originally we couldn't observe any difference in the average size of the adipocytes, when we were running an accumulative frequency distribution analysis, something that was calling to our attention is that both in the animals, in the MBO7 animals fed chow, as well as half-fat diet 45%, there was a higher frequency of smaller adipocytes. And this was in total contradiction regarding what we found in the adipose tissue when the animals were fed allele fructose and glucose, where basically we found a higher frequency of larger adipocytes. We are currently investigating in the causes of this, but essentially this is telling us that somehow the ablation of MBO7 in combination to the dietary stimuli is affecting the ways that the adipocytes and the adipose tissue are growing or expanding. We performed a battery of different experiments to assess how the ablation of MBO7 was affecting, or if it was affecting strongly, the adipose tissue metabolism. And here I am just showing two different experiments that we ran. And basically, the first thing is try to assess the adipogenic capacity of the stromal vascular fraction isolated from the adipose tissue of these animals and trying to differentiate them into full adipocytes in a plate. And this is something that we have reproduced systematically in an independent number of assays, where basically we observe that in the case of the EMBO7 heterozygote, there is a reduction in the adipogenic potential of the stromal vascular fraction. Moreover, in the mature fraction of the same animals that we were using for the adipogenic experiment, we were running a classical lipolysis activity assay as a classical assay that normally we use to assess adipose tissue dysfunction. As you can see here, when we were stimulating with different doses of noradrenaline, the capacity of the EMBO7 adipocytes were impaired. The lipolytic activity was reduced, okay, significantly reduced. Nevertheless, they were responding. The sensitivity to insulin regarding his antilipolytic action was totally preserved. Coming back to the transcriptomic analysis of our animal models, something that caught our attention is that we observe a very strong fingerprint regarding inflammation, similarly to what we observe in the liver of these animals. As you can see here, when the animals were fed the classical half-fat diet, we have a reduction in pathways related to inflammatory pathways. And that was totally opposite to what we found in the EMBO7 animals when they were fed allulose, fructose, and glucose, where the rest of the diets were providing some miscellaneous effects. Somehow this is telling us that the inflammatory component of the EMBO7 phenotype is something that we should consider not only in the context of the liver, but also in the context of the adipose tissue. We gathered some information using flow cytometry, where we observe some increase in the number of Kupffer cells in the case of the liver, as well as in the number of macrophages in the adipose tissue of the EMBO7 animals. So somehow this was driving our attention that we should focus a bit more attention to the inflammatory component. So basically what we did is that we were running some in vitro experiments using bone marrow derived macrophages as experimental model in response to different pro-inflammatory stimuli like LPS, chymosan, as well as lysophospholipids. And something that, before embarking in the difference that we observed, and something that was calling our attention is that we observed a substantial increase in the expression of EMBO7 in response to this pro-inflammatory stimuli. Our current interpretation of this is that in response to inflammatory stimuli, EMBO7 is activated to trying to reduce the amount of free arachidonic acid that is available for the biosynthesis of pro-inflammatory lipids. So somehow EMBO7 is providing a protective effect. Okay, so when we were checking how different was the inflammatory status of our EMBO7 macrophages, something that we observed systematically in these experiments with LPS, chymosan, as well as a different variety of lysophospholipids that are well known to induce the polarization to macrophages to M1, is that it was an attenuation in the M1 polarization of the macrophages, independently of the treatment. And we have combined with additional kind of treatments, and we see very similar results. So basically, we are interpreting these results as a potential interaction between the environmental and the nutritional milieu. And we should consider the crosstalk of the macrophages and the surrounding cells, both in the adipose tissue and in the liver, to determine the inflammatory status of the EMBO7 heterozygote mice. So basically, some of our findings are summarized in this slide. And basically, the first one is that the global partial ablation of the EMBO7 observed both in liver as well as in adipose tissue transcriptome affects pathways strongly related to inflammation, as well as cellular proliferation and energy metabolism. Nevertheless, something that was calling strongly our attention is that this pathophysiological phenotype was fundamentally associated to the dietary intervention use. Our results somehow are suggesting the importance of the crosstalk of other relevant metabolic organs like adipose tissue, pancreas, as well as muscle, as I haven't talked today about muscle, when interpreting the overall phenotype of our MBO7 animals. So basically, we are currently considering the possibility that the interaction between the genotype and the diet could explain some of the existing discrepancies in human studies, not only related to MBO7, but also in other US studies in relation to NAFLD and lipid-related genes, where the nutritional environment is not always considered or cannot be always considered. So basically, I am finishing here my presentation, and this is some of the people has been contributing to this project, and some of our collaborators like Matteo Resic and Tulia in Örebro University, the Newcastle University, as well as our strong collaborators in ABI, Brigham Institute, and our bioinformatic core facilities and lipidomic core in Cambridge University. And now I will be happy to take any questions further. Thank you very much. Basically, the clinical trials are providing evidence regarding the insulin status of the patients. But my understanding, for what I remember well, if I am not mistaken, when they are making the correction regarding body weight and insulin resistant status, they still find these associations of the ablation of the MBO7 in animal models. Basically, given that we see a reduction in the expression of MBO7 in genetically induced models of obesity, basically our interpretation is that MBO7 is somehow contributing to the NAFLD that we observe in all these models, of nutritional intervention, causing severe obesity or severe insulin resistance and type 2 diabetes. But at the moment, this is something that we are still trying to dissect further. No, we have done that. I remember that there is a paper related to LPCATs that is one of the other enzymes of the line cycle involved in the remodeling of phosphatidylcholine that I think they do some kind of work related to that, but we haven't conducted any kind of research to that. That will be very interesting. So I guess that she's making this comment in relation to the transcriptomic analysis. I don't know. We might need to go deeply into the transcriptome of these animals to see if there is anything that we can particularly grasp, because the number of pathways obviously have been cutting, the number of pathways that I have been showing today. Some of them might be related to senescence, so it will be nice to go and have a look. I mean, given that the expression of MBO7 is strongly reduced in the context of genetically induced obesity, I mean, it could be a factor that can discriminate maybe lean NAFLD. I mean, I don't have a clear answer to that. But one of the things that we need to consider is that we have been dissecting the action of MBO7 per se, but there is something that I haven't mentioned at all, because that will make our talk a little bit complicated, is that in the absence of MBO7, there is an increase in compensatory mechanisms coming from other genes involved in the land cycle, okay, to trying to accommodate the surplus of arachidonic acid into other lipid fractions. So somehow what we might expect to see here is a net result coming not only for the primary ablation of MBO7, but also all the compensation mechanisms that might happen. And this probably might explain the reason why we have some kind of mild phenotype in our animal models. But what you are proposing is something very interesting. It will be nice to explore that possibility.
And do you have any mechanism underlying that down regulation of MBO7?
I mean, Luca Valenti's lab has provided some evidence showing that insulin per se through PI3 kinase and FOXO are primarily responsible for this down regulation of MBO7. What we have done, and something that I haven't shown for the sake of clarity, is that we have been checking the promoter of EMBO7, and we have found a large number of clusters for LHR1. And basically, we have some preliminary evidence suggesting that phospholipids might increase, the isophospholipid might increase the expression of EMBO7. So we have the opposite. But regarding the downregulation, insulin so far is the only mechanism that has been proved to be responsible for downregulation. This is something that we started embarking in the lab, but we haven't continued this. This is something that we want to explore in vitro to see this profibrotic phenotype that has been observed in the other models, and see if somehow we can recapitulate that in vitro. We haven't done any of this work yet. Something that is intriguing me is this discrepancy between fibrosis and inflammation that has been observed in some of the preclinical models already published. Probably this is due to the fact that they are only knocking down EMBO7 in hepatocytes, and the fact that most of them might be using probably choline deficient diets. So in my opinion, they are using a more severe nutritional intervention to try to unravel the molecular mechanisms, or to have a better molecular mechanism for EMBO7 ablation. But yeah, what you are proposing is something that definitely we should go after. We haven't seen anything in the transcriptome, but maybe it's because I didn't pay too much attention. So this is something that I can come back to the transcriptome, okay, and in the different diets, and see if this is some of the pathways that are appearing there. Okay. Something that we didn't summarize here, because just for sake of clarity, is that there are a substantial number of unique pathways that are particularly activated or repressed under a specific nutritional intervention. And something that we need to consider is that despite we have been talking about controlled diets, every controlled diet that we are using has a unique fatty acid composition, and the term of control might be a confounding factor here. So every diet is somehow contributing differently to this pathogenic action of the ablation of EMBO7. So, and I think as despite we are considering the data globally, we might need to consider specifically every nutritional intervention that we have been using.
I think Antonio wanted to comment one of the Malu's questions before.
I was thinking that for me, the EMBO7 could become a biomarker if it helps us to prioritize some aspects of the diet. So individuals with a specific genetic makeup, whether they may be more with this type of information, whether you could try to design a type of diet that will be less damaging for them, or in some way may have a benefit. And I think what I've been discussing with Sergio at some point is how we could integrate the knowledge coming from these different type of diets in the context of the benefits or pro-inflammatory or anti-inflammatory responses, from the point of view that this, I mean, we don't know what is the right advice now, but I think should be possible to identify in the context of this type of genetic information, at least to provide some advice to avoid things that may be more toxic for these individuals. And I think that the differences in the phenotype that we observe in these animals with respect to the diet is very remarkable. Yeah. So I think this is probably the most innovative aspect of this, which I think is engaging on the links between specific macronutrients and specific qualitative aspects of the composition, and the response of, or the susceptibility of individuals with a specific genetic makeup related to EMBO7 for this.
I mean, in relation to what Tony is saying, one of the things that was calling our attention is that most of the papers that couldn't find any association between the SNP of EMBO7 and NAFLD are papers that are coming from Asian populations, Japan, Korea, Taiwan, so on. So despite, obviously, as I say, this is— So
Malu, that's the reason why we are in China. You know, we want to figure out that. We want to eat Chinese food and show that this is very helpful.
So basically, despite this can be explained by statistical difference, there is still the potential explanation that maybe the different dietary habits of these populations might somehow contribute to this absence of association. And I am unaware now if there is, not only in relation to EMBO7, but other NAFLD studies, if there are robust studies where they are also considering the nutritional habits of the patients, of the cohorts that they are studying. Because I think it would be very nice to discriminate the patients by the nutritional habits.
So the other thing I wanted to say is that EMBO7 obviously is hepatocentric because this is where the main insights are coming. But then I think what Sergio's data show is that other organs may be vulnerable to this, particularly under conditions of stress. And in some way, when someone has a mutation, doesn't mean that the mutation is only in the liver, it's everywhere. And I think in the context of positive energy balance, then could be some kind of effect that is amplifying the effect in the liver. And that is something that I think will be relevant to try to understand.
Thank you very much for being present today. And thank you, Sergio, for the excellent talk. It was really interesting to hear about these results, and we'll start paying more attention to the adipose tissue. So thank you very much. And I think if there's no more questions, we can end this meeting today. Bye. Thank you, everyone, for attending.
Thank you very much for inviting us.
Thank you.
Thank you. Take care.
Thank you. Bye-bye.
Bye-bye. Take care.